do a little joint presentation for us today. Josephine, thank you so much for being here, and I'll let you get yourself set up and then you can take it away whenever you're ready.
Okay, you can see presenter view, or what can you see now?
Yeah, it looks good to me.
Okay, so yeah, I'm sorry everybody. Jeremy was supposed to talk before me. He was going to talk about more generally biosecurity in Canada, and then I was going to take over for the end of the talk and talk about biological control. But he is struggling with time zone, so he didn't realize he was speaking now. So he's logging on at the moment, and hopefully he will be with us in a few minutes. But yeah, I'm going to take over for the beginning of the talk, and then we can rewind and he can start his part of the talk. Okay. So I'm going to talk about biological control. And so there are many types of organisms that we can use for biological control. You have these examples here of those types of organisms. Here are examples that we can use to control insect pests. The most commonly used organisms are predators, parasitoids, and nematodes, but there are also a variety of pathogens that we can use, such as bacteria, fungi, and viruses. And there are three types of biological control, namely conservation biological control, where the aim is to preserve the ecosystem and environment of the natural enemies, to make sure that the populations are large enough to control the pest. We also have inundative biological control, where the natural enemies are rid in the lab to build up the populations and then are released in the field. And then finally, the one that I'm going to focus on today is classical biological control. So classical biological control is an introduction of a natural enemy of exotic origin to control an alien pest. So we basically go into the native range of the alien pest, and we find natural enemies in the native range and then introduce them into a new area. So in essence, classical biological control is a controlled invasion. This means that studying classical biological control has implications for fundamental and applied research, since it will help us improve tactics for the management of invasive species, but also allow us to study invasions under controlled and predetermined conditions. So from invasion biology, there are three categories of factors that we know influence establishment and invasion success. First one is the invisibility of the area, which is basically how susceptible to invasion is the area of introduction. So is the ecosystem providing an available niche for the introduced insects? Are there going to be predators that could potentially regulate that insect? Or is there going to be competition for the niche? Things like that. Then there is propagal pressure, which is an umbrella term to describe the number of individuals introduced, the number of introduction events, and genetic diversity of individuals that are introduced from that species. And finally, within the factors that influence establishment are biological traits of the introduced species. And within insects, those biological traits that have received the most attention are life history, such as the niche, lifespan, size, characteristics such as social, solitary or gregarious, or even social. And then obviously the climate preference of the species. So now if we take those factors from point of view of biological control, regarding the invisibility of the area, we know that there will be an unoccupied niche for the natural enemy to occupy, since there is a pest we need to manage. In terms of propagal pressure, we can control the size of the introduced population. So now the last thing that we need to investigate is biological traits of the introduced species, of the introduced natural enemy. And one of the aspects that still require investigation is reproductive biology, or reproductive traits of those introduced species. And so this is where I specialize and what I'm studying at the moment. So again, from invasive biology, when we know, or from other fields. Rather, we know that reproductive biology affects various elements of population dynamics. So this can be like adaptation, inbreeding depression, sex ratio, population growth, valley effects. So we published a paper in 2021 that gathers evidence of the effect of reproductive biology on establishment in Hymenoptera species. And so for this talk today, I will be presenting the case of 4 species that are highly successful in their introduction range, and for which reproductive biology has had an impact on their success. So the first case is the case of Leptocybe invasa. It is native to Australia and is a gall-forming wasp that attacks eucalyptus trees. And from a reproductive biology point of view, it is a very interesting species because it has 2 reproductive strategies. A rare form of Leptocybe uses aronotoky, so it's a form of sexual reproduction. And then a more common form of this wasp uses tilitoky, which is a form of asexual reproduction. So here's the map of the range, the distribution of Leptocybe invasa. So as I mentioned, it's from Australia. Hi, Jeremy. And so it was first reported outside of its invasive range, of its native range in Israel in 2000. And between 2000 and 2014, it spread globally in most of the places where eucalyptus is planted like it's industrially planted. And so this success is mostly attributable to the fact that the populations that have been introduced and that spread like wildfire were reproducing asexually. And this is because tilitoky, or asexual reproduction, has 3 main advantages. The first one is that one female can potentially start a new population, contrarily to a sexually reproducing population. They don't need one male and one female, they just need one female to start a population. So this means that this reduces the necessary propagal pressure. But also, right after introduction, when the population density is very low, one factor that usually prevents establishment is the fact that females struggle to find mates to mate. And so for Leptocybe invasa, for the asexually reproducing species populations, there is no need for mate location. So that is not stopping establishment. And finally, again, because the females do not need to find a mate, so they don't spend time and energy with mate location, and they also don't need to spend energy on the production of males. This allows populations to grow very, very rapidly. So now the question is, if asexual reproduction is so advantageous, why would species even bother using sexual reproduction? And one of the main reasons for the maintenance of sexual reproduction across taxa and also within invasive species is adaptation. So here we have the case of Aphidius ervi, which is a sexually reproducing parasitoid. It targets many species of aphids and is native to Europe, but has been widely used in the Americas, Asia, and Oceania for classical biological control. And so here's an example of a study by Henrietta published in 2007. And so what they did is that they started 2 colonies of Aphidius ervi. One of them was reared on the pea aphid, and the other one was reared on the foxglove aphid for 50 generations. And then they measured the success rate of these 2 colonies on the two types of aphids. And what they found is that basically the colony that has been raised that was reared on the pea aphid was then more successful at parasitizing the pea aphid than the foxglove aphid, while the colony that was reared on foxglove aphid was more successful at parasitizing the foxglove aphid than the bee aphid. So what they showed basically is that when reared on different aphid species, Aphidius ervi can exhibit significant adaptation to new hosts in as little as 50 generations. That's about 3 years in a lab colony. That's very, very short. And that is possible because Aphidius ervi reproduces sexually and so is able to have a genetic recombination with each generation. Now, another way that there is another way that sexual reproduction can allow adaptation, and that's through hybridization between populations or species. And so here we're going to have a look at Apis mellifera scutellata and Apis mellifera. So Apis mellifera is native to Europe, has a high honey production, and is considered tame, and is also adapted to cold temperatures and long restricted periods, aka winter. And then we also have Apis mellifera scutellata, which is native to Southern Africa, has a low honey production, is considered aggressive, but is adapted to warm tropical and subtropical climates. So shortly after they arrived in the Americas, Europeans introduced Apis mellifera, so the European honeybee, for its high honey production. But as it was introduced in South America, the European honeybee was not producing as much honey as it was in Europe or in North America. So in the late 50s, and it was believed that it was because it was not adapted to high temperatures. So in the late 50s, Brazilian researchers decided to introduce the African honeybee into lab colonies in Brazil. And the goal was to hybridize, to create hybrid populations between the European and African honeybee to create a population that would produce high quantities of honey, but would also be tame, so less dangerous for honey production at high temperatures. But that's not what happened. The African honeybee escaped from the lab and then started spreading across South America very rapidly. And through competition, and because it was better adapted to warm climates, the African honeybee displaced the European honeybee very quickly in South America. Now, when the African honeybee arrived in northern Mexico and in the southern US, people thought that the range would stop spreading because the African honeybee was not adapted to cold climates. But what they didn't think about, or what they eventually observed, is that the African honeybee was starting to hybridize with the European honeybee to create what is now known as the Africanized honeybee, which is a hybrid population that is adapted to cold weather, like the European honeybee, but is producing lower quantity of honey and is also aggressive. So this population, or this hybrid population, is now considered also an invasive species, just like the African honeybee. So that's another example of how sexual reproduction can lead to climate adaptation. But for our last case, our last scenario, understanding the effect of reproductive traits on establishment is not only about sexual reproduction versus asexual reproduction, because these two types of reproduction can be done in many different ways. And that is why we're going to look at Paratrechina longicornis, which is a sexually reproducing species that has a very peculiar way of determining sex. So Paratrechina longicornis, as you can see here, is a widely invasive species. And what's interesting about this species is that the queens within the colony are haploid. So they only have one set of chromosomes. And when they want to create to make reproductive females, so more queens, they activate an unfertilized egg that is haploid, and this will then develop into a reproductive female. Now, when they want to make workers, they fertilize a haploid egg with a haploid sperm, and they create diploid females that develop into workers. But now the question is, how do they make males? Well, to make males, those queens create aneuploid eggs, so they don't have any genetic material from the mother. And those eggs are then fertilized by a haploid sperm, creating a haploid male. So as you can see, the only individuals that are diploid, that are carrying both the mother and the father genome, are workers, so they don't reproduce. This means that there is no recombination between the female and the male genome. What that means is that there is no inbreeding depression, because you cannot have an increase in homozygosity over time. So this is very valuable when individuals are introduced into a new area, because even if one female and one male are introduced, meaning that they're starting a new population, very, very low genetic diversity, this is not going to cause inbreeding depression. So that's very advantageous. The other advantage of this reproductive system is that when a queen arrives into a new area by herself and she cannot find a male of her own species to reproduce, she can do what we call sperm parasitism, where she can mate with a male of a related species and use his sperm to create hybrid workers. And because these do not really need to reproduce, their genome could be, if they needed to reproduce, would be genetically incompatible, but in individuals that do not need to reproduce, that's not a problem. So now we potentially have a population of workers that are adapted to the environment from the native range of the female species, but also the native range of the father species, which is the introduction range of the female species. So you now have very adaptable worker colonies. And this makes Paratrechina longicornis highly invasive. So to conclude with my part of the talk, we saw that reproductive ecology seemed to influence establishment and spread, but we still need more direct empirical evidence. And most of all, we need to understand how we can apply that to biological control. And this is what me and Jeremy are working on at the moment. Yeah, thank you very much.
So are there, I guess, not questions? I'll just go ahead. Is that? So can everyone see my screen?
Yeah.
Okay.
That's good, Jeremy.
All right. So Josephine just illustrated an important point, which is to always hire people that are smarter than you are. And given my snafu with the time zones here, it's obviously not that hard for me to do. So my apologies to both the Invasive Species Center and the audience for that slight hiccup, but we're on board now. So does someone want to remind me what time I should be done by?
You have about 25 minutes, Jeremy, so 1:20.
Okay. Okay.
And if you want time for questions.
Okay. Yeah.
You might also want to put your slides in presenter mode so you can flip through. And then you're good to go. Very good.
Okay. All right. So I'm going to talk today about forest biosecurity in Canada. And I would just point out that some of the ideas, or most of the ideas covered in here, are captured in a paper published with Mireille, Megan, and Todd in Frontiers in Forests and Global Change. This audience doesn't need to know or be reminded of this story, but you know, we're dealing with a large resource base, approximately 350 million hectares. primarily publicly owned, a big chunk of both our land cover and the world's forests. And perhaps the more salient point here, a lot of different ecozones and forest region types with significant economic impacts on the GDP in Canada and employment. This map here illustrates the enormity of the resource base and the variety of different forest types involved. And when we talk about biosecurity, what we're talking about really is an approach that encompasses policy and regulatory frameworks that's really designed for analyzing and managing risks. And so when we say forest biosecurity, we're talking about analyzing and managing risks to forest resource base. And so these two pictures here illustrate some of the consequences when biosecurity it fails, sorry. So the image on the left would be a spongy moth, or is a spongy moth outbreak from 2 years ago, I guess now, in the Muskoka Lakes region. This is my in-laws, the view from my in-laws' deck on their cottage. And what you can see here is that everything, and I mean everything at this time, was completely defoliated. And then the image on the right is borrowed without asking him from Troy Komodo. It shows some ash mortality in a woodlot in southern Ontario. And so these pictures really illustrate that these biosecurity these invasions, sorry, can have significant impacts both in terms of reduced growth, so that the trees shown on the left, they completely refoliated by the end of the summer and they're all still alive today. But it would be no surprise to this audience that that defoliation event would come with significant reduction in growth. And on the right, you can see significant mortality associated with emerald ash borer. And so these impacts, they have consequences for food webs. They influence microenvironments, obviously, nutrient cycles, successional trajectories are altered as a consequence. You get facilitation where invasive plants are facilitated often by association with other non-native species. And obviously, when you have mortality events, you get significant changes in coarse woody debris. And so these ecological impacts have political impacts as well. So Canada is a signatory on a variety of different international agreements. Some of them are shown here. And those ecological impacts have consequences for our ability to meet our requirements or our obligations as signatories on those agreements. So we have forest biosecurity being governed by both federal legislation, things like, for example, the Plant Protection Act, and certainly there are other acts involved, but as well by provincial and territorial legislation. So Ontario has Ontario Invasive Species Act. And the roles and responsibilities outlined in this legislation this Venn diagram is not an exhaustive depiction, but it begins to capture a couple of important points. And the first is that we do have overlap in the roles and responsibilities. So for example, you see Canada Border Services Agency shown in the far left here. They're responsible for enforcement and inspection. The CFIA also does some inspection. There are National Regulation and Policy Development Agency, and they manage incursion responses, but they also do some research, for example, which then places them in overlap with academia, the Canadian Forest Service, the provinces and territories. Another important point here would be that there's an iterative sort of feedback going on among the different players involved in forest biosecurity in Canada. So the Canadian Forest Service might, in response to a request from the CFIA, do research into a particular area. The results of that research might feed back into the CFIA, who might make altered recommendations to Canada Border Services Agency about enforcement and inspection activities. Similarly, Canada Border Services Agency might collect samples, submit them to the CFIA. The CFIA might analyze those, recognize a need for more information in something, and work with academia or the provinces and territories or the Canadian Forest Service to address those knowledge gaps. In forest biosecurity in Canada, when we think about the how it works, there's an invasive alien species strategy in Canada, and that strategy is predicated on 4 priorities. And I've emphasized here the first one, which is prevention. And the idea is captured in this graph that probably everyone has seen before. But the idea is that it's a lot cheaper to manage a problem if you never have the problem. So we're trying to prevent things from ever establishing. But if prevention fails, we then are emphasizing early detection. We want to detect problems as soon after establishment as possible. And that is because, as illustrated here, the economic returns on the investment in management are far higher the lower the population density or the extent of the area that's being targeted for management. We're trying to respond rapidly for the same reason, to try to deal with smaller population levels or reduced infestation area. And then ultimately, we're based on what we're seeing by in terms of population densities in the area infested, we're making decisions about whether or not to pursue eradication. And eradication might be pursued when you've got a small population level or a small number of areas where you've got established propagule. Transitioning into containment, perhaps you begin to have some of those areas coalescing, and the decision is made that we're probably not going to get rid of the problem, but now we just want to try to contain it. And then long-term control. And Josephine just talked about one of our major tools for long-term control, which is biological control. And so this slide here tries to capture the idea of prevention. So when we talk about forest biosecurity in Canada, there are those 4 priorities, but that prevention really is the priority that we're emphasizing. And so when we think about prevention, well, how do we do that? And so these heat map of the United States illustrates an important idea. So the first image that you see in the top left shows the number of pests per county in the area. And so as you go from yellow through red, through purple into blue, the number of pests are increasing. And what you see is a fairly clear transition towards the northeastern United States, where the number of pests are highest. And so then what drives that pressure or the number of pests that we see? And so then you're looking at also on the top right, the forested area in the United States. And what we see is that there isn't a really good fit between the area forested and where all the pests are, the highest numbers of pests are being detected. Then the 2 middle graphs show population levels. And so the one on the left in the middle is early 1900s. The one on the right is 2007 estimate. And we begin to see a better fit with where all the pests are being detected. So higher population levels, particularly as evidenced in the 2007 figure, correlate much better with where we're seeing highest numbers of pests. And then the bottom 2 graphics illustrate the economics. So it's an indirect measure of propagule pressure. So on the left, you see per capita income, and this is sort of an indirect estimate or measure of trade. And then on the bottom right, this is the distance to a major seaport. So really what these figures are trying to illustrate is this idea of propagule pressure. And so we need to think about pathways and how to manage those pathways to limit or reduce and prevent invasions. And this graphic, if you're not familiar with Kiln Digital, this is a data visualization company, and they do remarkable work. And so there's a lot of information here, but the points I would make based on this are, when we're thinking about pathways, this illustrates sea container movement globally for a window of time in May a few years ago. I can't remember when I took this data from, but if you go to their webpage, you can do visualizations and watch things and move it across or around the globe. And what you begin to recognize is that, one, there are lots of linkages. So if you look at Canada, you've got lines or vectors coming into ports in Canada from a lot of different places. And those linkages are not always direct. And so you have parts of the world that are connected to other parts of the world via, for example, say, Asia. So you have a container or a line moving, say, from Polynesia or Oceania into Asia, and then moving on from Asia to North America. And so this introduces the idea of bridgehead effects and the idea that something could be moving on container ships through one part of the world, ultimately ending up in another part of the world. The different colors illustrate differences in commodities moving around the world. And so It also illustrates the idea that we've got a lot of different propagule pressures. So not only are parts of the world connected directly and indirectly to lots of other parts of the world, we have a lot of different types of commodities moving along those pathways. And so what are the pathways that we're really worried about or interested in? Well, how do we get information on those pathways? And we primarily are using 4 different ways. So I've put in bold here participation in international networks, and a few of them that I've drawn attention to are things like International Forestry Quarantine Research Groups or Quadrilateral Collaborative Working Group, or things like the invasive species activities like we're participating in today, where information is exchanged and people begin to get ideas about what's happening in other parts of the world, because that's often a pretty good barometer of what's likely to come our way. We live in a global environment, and these problems, while the solutions are often implemented regionally, the way that we're going to ultimately manage these problems involves or necessitates the development and maintenance of international networks. But we also look at border interception records. So I mentioned that the CBSA is collecting samples when they see something, submitting it to the CFIA for identification. We do risk assessments. Various agencies are involved in that, and this involves, looking around the world globally and trying to predict problems that might be coming. And then, in particular, the CFIA does a lot of surveillance domestically. We're beginning to also do some surveillance internationally, where we do offshore mitigation, where we begin looking at both via sentinel plantings. So we plant or look at plantings of North American species that are planted abroad and try to get an idea of what agents are affecting those plants abroad. We also do lure or trapping activities abroad to begin to get an idea of what might be coming our way and what lures or traps might be effective if it does end up on our shores. So wood packaging is an important pathway, one that we worry a lot about. You begin to get an idea of the enormity of the issue. These images actually were given to me by Troy Komodo from the CFIA, and just looking at it, you see just how much wood packaging there is. And so this is obviously an important pathway. Wood borers and bark beetles can infest the area in the port of origin, and then the material is either put into a landfill as shown in the top left here, or containers are opened and insects have emerged or emerge once they get to the port of destination and then fly out into the environment. In response to this pathway, ISPM 15 has been developed, and this is the idea that we're trying to reduce the propagule pressure in this pathway, treating these materials before we use them, or trying to use clean materials to pack for wood packaging. We've also enacted a series of regulations like Plant Protection Act. We have an automated import reference system, AIRS, which is designed to try to reduce the live plant, the risks associated with live plant trade. But this pathway is become particularly complicated because now with the advent of e-commerce, it isn't as simple anymore. So now you have a much more challenging time trying to regulate that pathway, because material can be shipped by individuals or small businesses from different parts of the world. And this makes it a lot harder for agencies in Canada to manage this pathway. So border and post-border inspections, as I mentioned earlier, so at the border, this is Canada Border Services agencies. They're inspecting goods at these ports and land border crossings and airports. These inspection activities, clearly they can't inspect everything, so they're strategically deciding how to and what to inspect. This is informed by the risk assessments that I mentioned earlier and the historical data. The CFIA also does inspections that complement those activities of the CBSA. And post-border, it's really the CFIA, the Canadian Food Inspection Agencies, that's doing inspections at destinations of shipments, and they're doing surveillance within Canada. So our knowledge of these pathways, as I illustrated here, is based on border interception records and surveillance programs primarily. And the next portion of my talk, I'm going to focus on those border interception records. So along with the people mentioned for the paper that we published and somebody at Northern Forest Research Center, Bradley Tom. We're looking at the interception records from 2010 to 2019 given to us by the CFIA, and these are interception records both from the CFIA and the CBSA. So there's 14,000 interception records or specimens, and they come from a variety of inspection activities. Those activities, I'm not going to go through them here, but there's essentially 7 of them plus a catch-all, an 8th group that are called all other. And the identifications of those samples are done by some laboratories in Ottawa and Saskatoon. About 2/3 of those interceptions were identified to the species level. Some of them to the genus level. Based on the quality of the sample, sometimes it's not possible to go to that level, and we end up only identifying things to higher taxonomic levels. But when we look at that data, one of the things that really surprised us was we expected that there would be a pulse in the number of intercepts that would be driven by the time of year, that many of these organisms are only active in the summer months, and we expected then that to be reflected in the interceptions. But what we see is that, yes, there is a pulse, but it's not as profound as we thought it would be, that there's a fairly flat propagule pressure across the calendar year. And that's true both when you look at just numbers of intercepts, or if we take it rather to species. So you could imagine that you might get lots of intercepts of one particular species, but that wouldn't be reflected then if you just looked at the species level. But really what we see is fairly flat propagule pressure, regardless of whether we're just looking at total number of interception events or number of species intercepted. And so at first I thought, well, maybe this is a northern southern hemisphere thing, that if we break it out by where the trade is coming from, we might see more pronounced effects of time of year on that propagule pressure. And to some extent we do, but it's not as clean as I thought it would be. So you see a bit more, the blue bars being trade from the northern hemisphere, the orange bars being southern hemisphere trade. And so you would expect that, that sort of December, January, February, or October, November, January, February for the orange bars would be higher. And it kind of is. It's obscured a bit by the fact that the axes are biased towards the northern hemisphere because there's so much more trade and interception there. But we do see a bit of a pulse as well with the summer months for the northern hemisphere trade. But again, still fairly constant propagule pressure the entire calendar year for trade or originating from both parts, the southern and northern hemisphere. And then when we look at where those propagules are coming from, there are no surprises there. So there's a lot of trade within North America, and not surprisingly, both in terms of the number of intercepts and the number of species. A lot of that, those interceptions and species are coming from North America, then Asia, and then Europe for our dominant trade partners. So it really reflected trade. And when we look at what's coming, I'm not going to get into a lot of detail there, but that not surprisingly, some of these surveillance activities are biased towards insects, and insects being the most dominant organisms on the world. Given those two factors, it's not surprising that by far the dominant taxonomic group that we're seeing in the interception records are insects, followed by plants and then fungi. And that's true both for interception events and for species intercepted. And when we look at this crude map I've made here that shows the globe or trade coming from different parts of the world into Canada, unfortunately, the bars on the x-axis, the order of them changes a little, so it's a bit confusing to look, but it's the same groups. We're seeing insects as the dominant group, regardless of where the propagule is coming, or the trade is coming from, sorry, as well, followed by plants and then fungi. So when we begin to look at a little more detail, one of the things that is often looked at to predict establishment success, because there's a lot more interception events than there are establishment events, is that whether or not the host for the propagule, or in this case the insect, is present in the invaded or the area that it's introduced into. And so then we chose Lepidoptera because they have a fairly undiversified ecology. So all of them in this database, almost all Lepidoptera are herbivores. So we can look then at what the hosts are in the native range, and then look to see if either that species or a congener or a closely related species is present in Canada. And so the interesting thing is, as you would predict, if the host is not present in Canada, it's far more likely that that event will result in not being established than being established. So if the host is present, it's more likely that they will establish than not establish. Similar pattern, although less pronounced, observed with the Hemiptera, which isn't surprising because their ecology is a little more diverse than that of the Lepidoptera, but same trend that if the host is not present, that introduction event is less likely to result in an establishment than if the host is present. So while I would say these sources of information that we're getting are information, the information from these border interception records, risk assessments, surveillance programs, participation in these networks, I think they're incredibly important and we need to continue to invest in them. But I would say that they do have their limitations. So the images here are of an Ips typographus outbreak in Europe, on the bottom right showing all the dead trees, and then on the bottom left you see an individual Ips typographus. And I don't remember anymore, but it's over 400 times that this insect has been picked up or intercepted in North America, and to our knowledge is never once established. So while they're valuable sources of information, they're not perfect. And similarly, the other thing happens where something gets established, but it's never been picked up. The other caution I would point out is that despite the fact that a lot of these measures are very effective at reducing propagule pressure and designed to prevent the introduction of non-native species, despite their efficacy, you can still see an increase in interceptions and propagule pressure because trade is increasing. So although these actions are effective and they do have an impact at reducing propagule pressure, a concomitant increase in trade can result in an overall net increase in propagule pressure. And it's important to remember that even though we continue to see these events, it doesn't mean that our actions and our activities aren't important. It just means it's a very challenging thing that we're trying to accomplish.
Jeremy, sorry to interrupt. You have about 2 minutes.
Yep. Okay. So all of these things, as I said, so if prevention fails, we want to detect things as quickly as possible. We then wanted to have a rapid response to those invaders. So if that response or that detection happens around a port, for example, or on a commodity, then the CFIA is going to lead a surveillance to look for evidence of establishment in the area. If the detection happens in the environment, then they're going to do a delimitation survey to figure out the extent of the population they're dealing with. And the information that comes back from those surveillance activities is going to inform the decision if we're looking at maybe an attempt to eradicate, if it's a containment issue, and then we're looking at long-term control. And so I've already discussed the idea of that graphic and long-term control. So I'll jump straight to introducing the idea of these tactics. And we have limited tools in the toolbox, but if something does become established and we're looking at long-term control, biological control and tree breeding for resistance are really two of the dominant tools that we're exploring. And biological control is built on this idea that in the native range, an organism is limited in its population density by other biotic factors. And so by reestablishing the connection, so if that organism invades an area and is freed from those biotic relationships, we want to reestablish them to try to limit or reduce the population level. And so, you've got American chestnut, where we're selecting for resistance to chestnut blight. And here's a graphic that illustrates biological control. So you've got a high equilibrium or density of pest in the invaded range, where that arrow illustrates that the natural enemy is reintroduced with the hope that we realize a lower equilibrium for long-term population levels post that release. And that's my talk. I'm sorry that I was late again. I guess we might have a minute if there's a question or two. Maybe it's for Josephine.
Yeah, thanks, Jeremy, and thanks, Josephine. We do have a couple of questions. Maybe I'll do one quick one, and then you can answer any of the remaining in the question box afterwards. And then we've also put Jeremy and Josephine's emails into the chat if anybody has any questions for them afterwards. The first question, how much surveillance is being done? Are there people walking around container ports and trucking companies looking at most of shipments? Is this every day? Is this at night? Do we know what this looks like?
Okay, so maybe I'll take that one, Josephine. And that's a hard question to answer, Stephen. Certainly, you can imagine there's a lot of trade. And so, we're looking at a fraction of what comes in. And so a lot of those risk assessments and surveillance inspections are feeding information in where we're trying to make informed decisions about how best to invest that surveillance activity. So that the fraction of the total number of containers that are actually inspected, I don't actually have a good answer for you there. So I'm not going to hazard one, but I would say it's low. And that's by necessity. I mean, it's a daunting task to try to look at every single container. So there's certainly things we can do. So we know certain times of the year that the risk is higher. So, you do surveillance for a pest in Asia, for example, and we know that population levels are really high for a pest of concern. So then we can target inspections at that time of year on commodities coming from that part of the world. But it's a complicated question, Stephen. And sorry, Mackenzie, I'm just going to jump on. I saw Troy Komodo put in a question, how do you get LEPs established in an area without hosts? And so this happens all the time, and it's more common than we realize that herbivores are expanding their host range. And so you get organisms that show up in an area and they begin to attack novel hosts. And so sometimes that's predicted. So emerald ash borer shows up in North America and begins attacking North American ash when it's never done so before, but it has a history of attacking Asian ash, so it's not that surprising. But then you get examples where you've got plantation forests in the southern hemisphere where pines and eucalypts are being grown, and there are no trees that are even remotely closely related to those those areas in the southern hemisphere, and native insects are expanding their host range and becoming problems on those trees. So I think that illustrates one of the things we need to keep in mind is that yes, we can use biology to predict and inform our decisions, but we need to be aware of the fact that there are going to be surprises, and there's always going to be surprises.
Definitely, and I think that's a good point to end off on, and we'll maybe learn some more throughout the afternoon on some other forest issues going on across Canada. So thank you both, Jeremy and Josephine, for that great presentation. I'll cut you both loose for the afternoon. We really appreciate your time being here with us today. I'll give you guys a 5 or 6 minute break before we get started in our next session for the afternoon. So we'll see you back in the next session on Forest Invasives Research and Development Part 2 at 1:30. Thanks again to Jeremy and Josephine. Wonderful presentation, and we'll see everybody back at 1:30.
Thank you. And sorry, Mackenzie.
Oh, no problem, Jeremy. I know you're west right now, so.
Yeah, I was sitting there listening to it talk, and